So for those of you who are here this morning, I'm going to change tack more and talk more about how we can model epidemiology and evolution and address some of the issues about sampling and about how the details of the transmission process of an infectious disease might affect the phylogeny. So pathogen sequence data are often collected in many research and clinical settings, and those can be used to help understand the transmission dynamics of the pathogen. And a lot of this has been driven by data. So there are over 1 million sequences in GenBank, and around half of them are SARS HIV. Next, influenza A, then hepatitis C, hepatitis B. Here's a typical virus. This is an old slide. There are about 16,000 norovirus sequences now. And then everything else. And there are about 3,500 genera recognized in GenBank. So this is divided up by between lots and lots and lots of taxa. So it's not surprising, perhaps, that these three viruses really have driven development in so-called phylogenetic studies. So there's been progress over the past 20 years, as well as, I think, obstacles. Firstly, although there's a huge amount of data, we can't use all of that data productively because of the way that it's sampled. It's often using convenience sampling rather than some probability-based sample. And that limits the confidence with which we can draw conclusions. And despite many methodological advances over the past 20 years, we're perhaps not quite where we should be. There's a number of reasons for that. Firstly, epidemiologists often think of things going forwards in time. You think about predictions, you think about time from the present going forwards. Whereas evolutionary biologists think about the present and they look back into the past, what happened in evolutionary terms. And those different perspectives, I don't think, really have helped the melding of evolution and epidemiology. There's also a fair amount of inertia relating to models that go beyond the standard coalescent model that's a cornerstone of population genetics in a phylogenetic context. That coalescent model was developed in the context of a large population changing deterministically. And it has lots of nice properties. Perhaps the nicest, and why it was so popular, is because you only have to consider the evolution of a sample of sequences. You don't have to worry what's going on at the population level. And in the early 80s, when the coalescent was formalized by John Kingman, the fact that you only had to worry, say, about 10 sequences rather than a population of 100,000 or 1 million was very important, especially in the context of what computational facilities there were at the time. However, because of the way that one has to scale from the sample to the population, and because of the desire that multiple models, including those that have discrete generations, also scale to this coalescent process, that restricts the sort of models that it can deal with, particularly in terms of selection and in terms of sampling fraction. But those really are restricted to models that have discrete generations, and most epidemiological models are in terms of continuous time. So if we're prepared to think about things in continuous time and not worry about generalizing to discrete generations, and if we're prepared to worry about what's going on at the population level and consider the dynamics at that level as well as what's going on in the sample, then we're free to do pretty much anything we want to in terms of inference and simulation. Computational issues still remain, though, and we try to do the best we can. And it is a multidisciplinary problem, and depending upon how you view that, this can be a challenge to try to get different disciplines together, or an opportunity for collaboration and opportunities without conflicts. So what we would like to know, for example, is we have a transmission tree where individuals may become infected and then recover or die and then transmit to others. But even in the most ideal circumstances where we can reconstruct the times of these branches. If we have a phylogeny, we lose a lot of information. So we lose the common ancestor, we lose the direction of transmissions, we lose all the individuals who were recovered before our sampling time point, and we probably don't sample everybody at the present either. But despite that, there is still a lot of information. There's information on the incidence and prevalence that we can get from looking at the lineages over time. We can look at clustering of sequences from different subpopulations, and that tells us about population structure. As Sebastian Bonhoeffer described a couple of weeks ago in the workshop, imbalance in the tree, asymmetry in the tree, can be suggestive of hidden population structure. At the ultimate level, the tree topology and the branch lengths does provide information on who infected whom, although one has to be very cautious with how you interpret that data. And finally, and a real challenge, is trying to look at recombination. Now, recombination causes all sorts of problems trying to estimate a phylogeny, because now you don't have just one phylogeny, we have different phylogenies for different parts of a region. But if we can try to specify models for recombination appropriately, then that opens up some new opportunities, because each of those segments represents more independent information about the transmission process. Or we can start to learn things about multiple infection from those data. So to formally link epidemiological models with evolutionary ones, we require these, the P word, as Ollie likes to call it, phylogenetic models that combine elements of both. There are a number of challenges to that, though. Firstly, there is this issue that we think about when we write down, say, an SIR model, we think about time going forwards. But most coalescent models consider time going backwards and in different units. So epidemiological models are in terms of calendar time, but coalescent models are framed in terms of generations. Also, a typical epidemiological model, even the simpler ones, are much more complex than the toy models that underpin coalescent models. And sampling issues are very rarely considered. So a standard approach for trying to estimate a so-called effective population size over time is a thing called the time-varying coalescent. The idea is that we have the number of lineages we call A, and as we go back in time, they drop off, they fall, and they decrease at a rate that is determined by this so-called ancestral size function. And the smaller this ancestral size, the more rapidly the lineages die off. And this relates back to a population genetic concept proposed by Sewell Wright, that if you take two random individuals in a big population, we have to go back many generations before those two lineages coalesce, whereas if we take two individuals from a small population, we only have to go back a small number of generations. That's just founded, this is a generalization of that. So I'm going to focus mostly on numerical simulations that of models that combine epidemiology and evolution. You can get some analytical results, although those are usually for somewhat toy scenarios. And so the basic concepts, I'll talk about some differential equation modeling of how we can incorporate things that we can get from phylogenetic trees and how we can incorporate that into a classic compartmental framework. And that's work with my former postdoc, Eric Volz, who's just started at Imperial. And I'll also talk about similar models, but stochastic rather than deterministic. And this is with my postdoc, Lorenzo. So the most basic information we can get from a phylogenetic tree are the number of lineages over time. So if we go to this transmission tree, and we've sampled these 6 individuals, then the number of lineages goes down over time from 6 down to 1, where we lose any more information. But along this, there are a number of invisible transmissions, ones that we don't see because we didn't sample both lineages here. So there is an invisible transmission here because this person recovered. This person infected, person 3 and person 2, but because we didn't sample person 2, we don't know about this one. We just had to guess that it was there. It turns out that at least for simple populations, whether a coalescence occurs in a tree depends upon the following rather simple recipe. Does a transmission occur? If there are multiple populations, subpopulations, between which subpopulations does a transmission occur? And if there is a transmission, do we sample both lineages involved? If we don't get a coalescence. If we do, we do. So let's take a toy model of HIV with a susceptible and an infected class. This is an open population, so there's input of susceptibles. Individuals become infected in a mass action fashion, and there's additional mortality term. So that's the simplest possible model of HIV that has any bearing on reality. And what we can do is, rather than just run that forwards, we can run it backwards. So let's say we've run our equation forwards, and then we can look back in time and say, what was the infection rate? And that's really just the beta Si over n term. And also, how many individuals of those I individuals infected at a time did we sample? So then, this represents the possible transitions that we can have. So if we have sampled 2 lineages, both of them, then if there's an infection event, then those coalesce down to 1. And that occurs at the transmission rate, and then the probability of sampling both of those lineages. So it's just a fraction, more or less, of the number of lineages divided by the number of infected. U's represent the other individuals, the uninfected individuals we didn't sample, the infected individuals we didn't sample, sorry. And if there is a transmission event between 2 individuals where the lineages didn't end up in our sample, then this doesn't affect the number of lineages at all. So although there are a number of different things that can occur, depending on whether we've sampled or not, ultimately everything boils down to whether coalescence, whether a transmission occurred, and then did we sample it or not. So this means that we can model in a deterministic fashion, so approximating a sample of lineages by a differential equation. So let's say we've got lots of sequences. We can model the number of lineages going backwards in time now. I'm going to denote backwards time being S, simply as a product of the infection rate and then the proportion of lineages or sequences over the infecteds squared. So this is for a large sample size. So that can tell us a number of things. Firstly, it can tell us what that effective population size is in an epidemiological model. And there are 2 equivalent but distinct interpretations of that. This is discussed a bit by Trevor Bedford's talk a couple of weeks ago. There's a population genetic interpretation, and that is that the effective population size is a product of the number of affected individuals. The more there are, the bigger the population size. The generation time, as well as the inverse of the variance in generation time. There's also an epidemiological interpretation that is mathematically equivalent, and it's the incidence over the prevalence squared. So why does it make a difference? Why is it important to give an absolute value for this effective population size? Well, taking that toy HIV model and running it over time, this is on a log scale, And here are the number of infected individuals going up to a peak and then declining. And then the dashed line represents this effective population size. And even though it's a fairly simple differential equation, it matched extraordinarily well to stochastic simulations. This is in a population, typical population of 10,000 individuals. What's important to note is that the timing of the peak of the actual population size and the timing of the peak of the effective population size do not coincide. And that's because this is a product of the non-infected individuals, this, as well as the generation time, which starts off very rapid when the susceptibles aren't limiting, but then slows down a lot as the susceptibles are used up. So what happens is that even though the number of individuals is declining, the generation time continues to increase. And so there's not a peak. These peaks don't coincide. So what many studies have done when they've done phylogenetic analysis is to look, say, at where these peaks are and put interpretations about the conditions that were going on. What was the epidemiological context of this peak? What they're really talking about is what was happening here instead, what they should have been talking about. There are other difficulties with generation time, apart from the fact that it doesn't remain constant over the epidemic, as is usually assumed in a population genetics model. So because we consider time in generations going backwards, there's a difference between that first coalescence, or first set of coalescence events, and the next ones. Because the last coalescent event in a tree is actually right-censored by the sampling time. It's not closed at all. So what was pointed out by De Silva, Christoph Fraser, colleagues, is that if you actually fit a model, a nice flexible parametric model, to many epidemics that are actually truly exponentially growing, you get this artificial tailing off of the epidemic. And that's just because there is this censoring at the very end that these models do not take into account. And one can imagine circumstances where epidemiologically that would be very important to know whether a disease was still exponentially growing or whether it's tailing off. There are many other things that we can get from a tree. One of them is to do with the shape of the tree. So here is a very symmetric tree of 6 individuals. See, it's perfectly balanced. And here is one that's more asymmetric. So this looks a bit like an HIV tree, and this looks a bit like a flu hemagglutinin tree. And we can capture the level of imbalance of that tree by a number of statistics. So a large number have been proposed. Here are just 2 of them. One is to look at so-called cherries. These are tips that share a common node, immediate node here. So there's 1 cherry here, 1 and 2, and there's 1 cherry here, 5 and 6. In an asymmetric phylogeny, you get fewer cherries, just 1 here, 5 and 6. There's also another index of asymmetry that is simply based upon the number of internode distances as we go from the tips of the root to the tree. So here it would be 1, 2, 3, 1, 2, 3 for number 2, only 2 for number 3. If you add all those up, that gives you a number that's called Sakin's index. And if the tree is more asymmetric, Sakin's index ends up bigger. So we can capture asymmetry numerically using those sorts of simple measures. So this approach was considered by Gabriel Leventhal, and who ran a number of simulations and found that under a number of different models where individuals had different contact rates, so these are different graph models, Erdos-Remy, Watt-Strogatz, etc., that you end up having more asymmetric trees if there are differences in the opportunities of individuals to become infected. You can see that in lots of other data sets. Here is an early data set of HIV from the United States, sampled in 1992, so before widespread therapy. So this is essentially all unaffected by therapy. And you can't see very much here, but when you calculate asymmetry, the observed value is here. Here is the null distribution by a simple permutation test, and it's significantly more asymmetric than one would expect by chance alone. Here's another data set of HIV, this time from London, from Chelsea and Westminster Hospital. This is a larger data set of 1,600 or so sequences. So what we can do is we can take a sliding window going from the earliest sequence to the latest sequence, to a window of 500 individuals, and then we can calculate this asymmetry measure. And as we go from the old sequences to the new sequences, we start off having a very asymmetric epidemic. But as we get later on into a more endemic phase, then the asymmetry tends to go away. And we only really see this with Sakin's index. We don't see it with the number of cherries. That remains fairly constant, a little bit significant, but not much. So this is really structure that's driving asymmetry very, very early on in the epidemic, and it tends to go away later on. So what can we get from that? What can we conclude is actually driving that asymmetry? Leventhal et al. Said that was something to do with things like network structure. But pretty much any statistic that we can think of that we can derive by post-order traversal of the tree, we can calculate and put into this differential model framework. So let's divide the branches of the tree into two types. One's at the tip of the tree, and then internal branches. So when two tips coalesce, they make a branch. When a tip and a branch coalesce, they make a branch, and so on. And when two tips coalesce, that makes a cherry, just based upon the definition of what a cherry is. And that simply occurs at a rate which is the infection rate and the number of tips over the number of individuals squared. So we can calculate lots of things. I won't bore you with how these things are derived, but we can work out Sakin's index as well, just by summing up the number of coalescences going back in time. We can look at clustering, we can look at population structure, lots and lots of things. And the nice thing is that you can take your favourite deterministic compartmental model, if you are so inclined, and you can work out what the tree should look like. So you can get lots and lots and lots of statistics from that. In a very simple sort of plug-and-play sort of way. The nice thing is that because we have a very low-dimensional system, just a few differential equations, and because we can rapidly explore how processes affect these, we can then reverse engineer data sets and use them as a basis for model fitting using approximate Bayesian computation. So I won't go into that further, but it seems to be quite a useful strategy. This is a comparison of the differential equations for different statistics that we can calculate from a tree, just based upon simulations. So the number of inches over time we can capture very well. This is the distribution of the length of the tips of a tree. This is if we look at a tree and then say we're going to call two sequences clustered if they share a common ancestor within a certain period of time. So it starts off with nothing being clustered and going up to large clusters. We can calculate the number of cherries and Sakin's index. We do a remarkably good job in capturing those dynamics using very, very simple equations that are more or less just binomial sampling and a rate coefficient. Things become a bit more complicated when we move to a structured system. It's just a bit more bookkeeping, but the same principles apply. We have to think about the rate at which infection occurs and then how many lineages of each type we have. But again, it's just rate coefficients divided by binomial probabilities. So, where am I going with this? Well, Leventhal et al. Assumed that heterogeneity arises solely due to variations in contact rates. They didn't consider any other alternative competing hypotheses. But there are lots of potential sources of heterogeneity. One is that individuals are typically much more infectious during acute HIV infection. There are also big differences in infections between individuals because the viral load within their bodies varies by orders of magnitude. There might also be different susceptibilities to infection. different hosts. And so that is just a valid an explanation, at least qualitatively, for the asymmetry that we see. And what this framework allows you to do is to be able to assess whether biologically those things are reasonable explanations. So I took a model that had acute infection, where you had a 3-month period of elevated infection, about a 20-fold increase in the infectivity parameter. And so you initially see, this represents acute individuals and chronic, and the epidemic starts off being very asymmetric to begin with, and then goes away. We can also look at to what extent acutely infected individuals cluster with each other in the tree. It starts off being very clustered and then goes away. And we actually see values of asymmetry that are not inconsistent with the real data. However, if we do the same thing, but we now include a simple sort of core group model, where we have a group with a high contact rate and a low infective rate, and a variety of different mixing structures, we can't really get the levels of asymmetry that we see, say, in the Leventhal data set or on those other 2 data sets I showed you. That's partly because if we have, everyone goes through acute infection, so that contributes quite a lot to asymmetry. Whereas with a core group model, we either have a small number of individuals, so they don't really contribute very much to the tree shape, or we have a larger group of individuals and then the population is more homogeneous. So it's very difficult, at least in our hands, to come up with a simple core group model that can explain the levels of imbalance that we see in real HIV trees. We can also get into somewhat dangerous territory by looking at who infected whom. And this is a common use, at least in a heuristic sense, in court cases where one has a phylogeny and you might have an individual that has been accused of infecting another. And the models can be used to estimate the probability of who infected who. So, so if we assume a model, and this is a biggie, for the transmission of HIV at the population level, we can estimate the probability that 2 people in our sample form a pair linked by direct transmission, as well as the direction of transmission. So you think that this might be somewhat magical. It's actually not magical. It just formalizes what we kind of already know. So, if there is a homogeneous population, we can't infer the direction of transmission. So if you have a pair of individuals, the probability that A infected B is basically a half, because we don't know which way around it goes. However, if you do have information, for example, one person has been infected a week ago, another person was infected 6 months ago, then you can use that information to get some idea of what the direction of transmission might be. So the simplest possible case that you can get some sort of analytical insight is with these cherries. So if you have a tree and you see 2 individuals that are closely related, it's tempting to look at those and think, well, perhaps those represent direct transmission. So we can formalize that and see to what extent that might be true. That all depends upon, in that cherry, whether there were any invisible transmissions in either branch of that cherry, which obviously depends upon how distantly related those 2 individuals are. So, the rate at which an invisible transmission occurs is essentially this here, which essentially that shows that there is an infection rate, but it can't end up in our sample. But again, the form is very, very simple indeed, and it's just a function of the transmission rate, of number of infected individuals, and our sample size. So this represents, in that simple HIV model, if we have a cherry and we know we can put a timescale on that, we can say that the common ancestor was, say, 500 days ago, we can work out the probability those 2 people were directly infected. And so here it's about 20%. Obviously, because this is a simple model, totally homogeneous, the best that we can do is come up with a probability of 0.5, because we can't get the direction the way around. So my initial concerns about this being used for evil doing and so on, I think were somewhat unjustified. What it does show is that it does formalize how difficult it is to to be able to conclude direct transmission from phylogenies. Our sampling is almost never good enough to be able to say anything with confidence. This also relates to an issue of reproducibility. Many papers don't publish their full sequence data of HIV because there are fears that you might try to reconstruct transmission events. And if the sampling is such that it's reasonably random, then the chances that you are going to find infected individuals where there are direct transmissions is very, very low indeed. What you can do actually is to use these models, identify those individuals and then trim those out. So you can release the maximum amount of data that you can, but still be fairly protective over individuals. In fact, anything that you do, this is a very conservative estimate of transmission. So this shows you the proportion of cherries that represent direct transmissions compared to all of them for different ages of cherry. So the maximum number of cherries you can get in a tree are a third of all the samples, so 0.33. If we have only 10% sampling, only a small proportion of those cherries represent direct transmissions. But even if we have 90% sampling, very, very unusual, only about 2/3 of those cherries actually represent direct transmissions. 1 out of 3 don't. And that's way beyond reasonable doubt, I think. And you can do so a little bit of calculus to work out actually what this number represents and what it's a function of. And it's actually a function simply of the number of affected individuals and your sample size. So there are more complex models though. It's not just individuals who fall into cherries, like here, that might represent transmissions. It could be that you have a third individual who infected both of those. Yeah. Or it could be that J infected I, who then infected K, and so on and so forth. And that's maybe even more difficult by incomplete sampling. And so Eric Volz has done some nice work on this to show that the probability that one individual in a sample infected another, you can do that for all pairs of individuals in the sample, not just those in cherries, as well as for quite complicated models. So I'm going to wrap up with two things. Two challenges, stochastic modelling and then recombination and co-phylogeny. So all the work I've talked about so far uses differential equations to capture the structure of the epidemic tree. But in almost all cases, when you look at phylogeny, they all collapse down to a common ancestor, and probably, at the time of that common ancestor, the non-infected individuals is probably quite small. And so the assumption of a deterministic model that might be appropriate now is probably not the case early on in the epidemic. Also, stochastic models are arguably better for inference purposes, at least to be nice and robust. In all deterministic models, it's very easy to play back time in reverse. So we just run the thing forward, and then we take the output of that model and we feed it in as input to another ODE system. But it's not as straightforward for a stochastic model. So running stochastic models forward is a well understood problem. The idea is we can formulate things mathematically as a probability of being in some state x at time t, given that we were in a former state x0 at time t0. And we can simulate, this is normally a big state space we have to worry about, so we can't solve that problem in general, but we can simulate from it. And we can do a lot of the simulations even though it's not as satisfactory. And this goes back to at least Gillespie's algorithm in the 70s, that if we initialize the trajectory, we work out when's the time of the next event, and then given an event, work out what it is, and do that iteratively until we get to the final time. So it's all pretty straightforward. So we start off with an initial state, and then we go to the next state. So that causes problems, though, if you want to have these coalescent models, because what's the time that we have between the first infection and the most recent common ancestor of the sample? How do we model that? Also, how do we constrain the simulations that are going forward in time so that they end up having the right number of sampled sequences, especially considering that The sampling process might be quite complicated. So Tim Vaughan, working with Alexei Drummond in 2010, proposed an algorithm to simulate stochastic models going backwards in time. And this is a bit more complicated. So you start off with a simulation that's very much like the Gillespie algorithm. You generate a new time step, but then you either update the trajectory, you then copy the current trajectory, or you delete and start again. And this actually is a hack for normalizing the likelihoods to become probabilities. So it's actually a very clever system. And so this is what you do. You have your state, and then you try to make a state earlier in time, and you either do that by itself, or you make a new trajectory plus an old one, or you ditch that trajectory. So this works pretty well. So we've taken this approach and we've run epimodels backwards. And it works very well, as one can imagine, with things such as the separate infected model, because then you just have infections clocking up. So playing it backwards is very, very easy. For the SAR model, running it backwards is a bit more complicated. Although there is this sort of, there's no quasi-equilibrium state, actually specifying what a good initial condition could be, both infected and recurrent, is difficult. I do, yes. It should be. There you go. See if that does anything. So for the SIR model, it might be difficult to specify the initial conditions. And for a SIS model, then the system has lost memory of when the epidemic started. So we're excited about this approach to begin with, but at least for models that have any sort of equilibrium behavior, it doesn't seem to work that great. So if we start off with an SIS model, and we start here, then we gradually drift back over time to a lower level. The equilibrium's up here. If instead we, and so this represents, the black line represents the numerical solution of Kolmogorov-Blackwood equation, and then the blue lines represent our simulations. The idea being that for more complicated models, we probably can't get this black line. So we do a pretty good job. This represents what happens if we start off at equilibrium. And the model doesn't know that we had to start off with a small number of individuals. It thinks, oh, we might start off with 100, and then gone down to 50. So at least just in terms of simulation, rather than in terms of inference, these models aren't particularly useful. So what we do is to overcome the problems of getting good prior distributions and ensuring that the sampling is matched. We employ a dual approach, where essentially we simulate the process forwards in time, and then we go back and we label each of those transitions according to whether they were sampled or not in the phylogeny. So we can go a bit further than that, and we can actually use these simulations as the cornerstone for inference. So for the simple SI model, then if we sample all infected individuals, that phylogeny tells us the time of all the infections apart from the first one. So if we condition and don't worry about that, we can write down the likelihood, and we can get estimates of things like the population size and the infectivity parameter just in the phylogeny. You can write it down. The problem is, of course, there are these invisible transmissions which arise due to incomplete sampling and so on. So simulations can be used to fill in the gaps in the data, and then once those gaps are filled in, we can then work out what the likelihood is and get the estimation of the parameters. So the question is, well, how do we actually get simulations that match our data? So we have to constrain the simulations somehow so that they match the sampling as well as the coalescent events. So what previous studies have done is they've taken time and they've chopped it up into discrete sections. And that way they can enforce that coalescence occurs at a particular time step, and then they can write down the coalescent. They can write down the likelihood. But they still use these large matrix exponentials, so they have to use a number of approximations for that. Other approaches have assumed a very simplified sampling process. So there is a SIR type model based on a birth-death process, but it assumes that as soon as you sample somebody, they can't infect anybody else. And that might be okay for flu, but it certainly isn't the case for things like HIV and hepatitis C. So I've taken an approach that suggested by T.J. McKinley, it has a nice paper, it's out now, that rather than looking at the time to the next event and then deciding what the event is going to be, you ask, does an event occur before the next time point that I fixed? That's the sampling time or the coalescence time. And then, if so, when does it occur? So this way we can simulate these invisible transmissions, and then any biases that we have in the simulation process we can correct for using importance sampling. So here's an example of how this works. So in black represents the number of lineages that we see in this model, and red represents what's actually going on at the population level. So there are more infections here, but the times of a subset of the infections are shared. There are other ones that occur in between that we don't see. So essentially we can fill in these gaps, and actually because this is a simple Gillespie type simulation, we can just marginalise over those and get estimates of things like the infectivity parameter, and do so in a way that avoids approximation by discretization of time. So the last bit is on recombination, and everything I've presented so far assumes that there is a single tree. And recombination, however, is rife in many viruses and can lead to different evolutionary histories in different parts of the genome. So a number of years ago, Sergei Kosakovsky-Pond and I developed an algorithm to look at recombination in a sequence alignment. The idea being that if there's recombination, different parts of the genome support different trees. And the algorithm uses a genetic algorithm approach to chop up the alignment into segments, fit trees to different segments, and then work out whether that gives you a better fit than having a single tree alone. So here are some simulated data where we have trees that alternate between two different topologies, and we can estimate where the breakpoints are and what the topologies are. We actually do a remarkably good job. So we can take this and apply it to real data. So here are some data sort of hot off the press from a study of HIV in Beijing. It's in collaboration with Sten Vermund and Marsha Kalish at Vanderbilt. And these are a small number of sequences taken from individuals who have a combination between two different viral subtypes, B and C. What our algorithm can do is say not only that they're a mixture of different subtypes, but where those breakpoints are. And just eyeballing it, one might get the impression that these sequences are all the similar recombinants. This one here is very similar, apart from there's another recombination point here. So perhaps these viruses are related to these ones. Then there are ones that are very similar, but this one segment's been swapped out, and so on. Of course, we don't just have the subtype structure, we have all the variation within those segments as well. So using these sorts of approaches, we can get a very detailed picture of not just a tree, but a whole network of transmissions occurring through a population in a way that would be virtually impossible with classical epidemiological means. However, dealing with that sort of recombination, apart from just describing what the patterns look like, is difficult. It might be difficult for other people, but it's difficult for me. So, as a baby step towards this, we're considering so-called co-phylogenetic approaches. These are approaches that stem in looking at hosts and parasites, with the idea that the phylogeny of a host might impose a phylogenetic structure on the parasites that infect them. So we're applying this to retroviruses in red colobus monkeys in Uganda. We're applying this to mobile genetic elements in bacteria, as well as in different influenza segments. So here's a description of one part of this big study that we have on retroviral transmission in western Uganda, and the wildlife part of it centres around red colobus monkeys that are quite populous in this area of western Uganda, but they're absolutely chock full of viruses. glorious one. So we've got 3 different retroviruses at least, as well as a newly characterized hemorrhagic fever virus. And these viruses, even though this is an endangered species, in the local area, this is the #1 animal you come into contact with. Not dogs, not pigs, anything like that. So you get most bites and that sort of stuff from these animals. And full genome sequencing has found that the red colobus up here has 2 distinct co-circulating hemorrhagic viruses. And actually these red-tailed guenons that hang out with the red colobus also have their own variants, own hemorrhagic virus variants. And these are very, very different than each other. Originally hypothesized they'd share variants because they hang out together. So these live in the ground, these live in the trees. So the red colobus look out for eagles and so on, and these look out for snakes, and they warn each other about predators. So that's one hypothesis about why they forage together. So this is based upon full genomes of this new hemorrhagic fever virus. This one variant, so this is 2% divergence. We've got quite a lot of structure there. We've got some long branches. We've got some very close related viruses. And this is the other phylogeny. What we can do, we can place those phylogenies end to end. This is at the top. They've also got SIV that bears no relationship to the transmission of this hemorrhagic fever virus. And there are a number of clades that you can see that actually join up together. So although overall there's not a great deal of shared topology, these red dots represent phylogenetic structures that are shared. So it's very weird that 2 close related viruses can actually co-circulate, co-infect the same individuals. It's also kind of weird they seem to be co-transmitted. What we need to do is be able to put the same timescale on both of these and allow the phylogenies to be at least partly constrained in order to come up with an idea of how often co-transmission occurs or how often one transmission occurs after the other. That's the next step. So not so far off, I think, is stochasticity. We're working on it. David Rasmussen, Katy Koehler has some nice stuff, hopefully impress on stochasticity. So I think we'll be there, at least in terms of inference, not so much in terms of analytical results and stochasticity. I think structure as well, haven't spoken very much about it, but at least in terms of compartmental models, not so much in terms of networks and things. And that's still very far off. Still some time away, understanding how recombination works, which does make a huge difference, but also selection. Many of these phylogenetic approaches are applied to influenza, which undergo immune selection, and potentially some variants are more transmissible than others. But it's not clear to what extent selection really does distort the phylogenies. Still compared to a model that we fit that doesn't assume selection, but assumes some sort of equally variable infectivity. So with that, I'd like to thank my various collaborators, as well as funding from the Royal Society, Wellcome Trust, MRC, and NIH, and ESRC. Thank you. Any questions? Or is everyone sick to death when you talk? Yeah.
In the face of recombination, are the trees so powerful that they're worth hanging on to? I mean, could some other clustering approach work better?
If recombination was so rapid that linkage was completely absent, then the frequency of the different alleles that we see at each position would be very, very informative. So the other extreme of no linkage is also great to work with. Unfortunately, we're working in this rather horrible area where there is some recombination, but not so much that we can assume lack of linkage, not so little that, at least as we go to these full genome studies. So it's fine, when you just sequenced 300 base pairs of HV envelope, you didn't have to worry about recombination. There wasn't enough opportunities there. Now we're getting full genomes, we see it all over the place. And also, as the epidemic's progressed, we see more divergent isolates, and so our power to be able to spot it. And I don't think that people who do these analyses will be able, myself included, will be able to ignore recombination for much longer. So the standard approach is just to ignore it and say that it doesn't really affect inferences.
How much of this can actually be applied to endemic diseases? Because most of what you were showing were diseases that were in epidemic progress, exponential growing, and for them it's very obvious that if you move backwards in time in the epidemic part of it, it's actually a stable process.
Yeah, you can get some nice results from that. So it actually makes life much easier. So all the analytical results that I've generated assume a stable population size. But actually, if you look at the time scale that it takes to go back to that common ancestor, it's usually very long. Usually much longer than an epidemic could realistically be around for many of these diseases that we're focused on. So the coalescence of influenza might go back to, say, the last big epidemic, or HIV might go into the introduction, might go back decades, but so you can get some neat results out of the stable models, but it's not clear how applicable those are to real data, just because the timescales, you almost always have to go to the dynamic regime.
How related is the distribution of branch length, the generation time distribution?
Do you mean like in terms of non-exponential?
So no, I'm saying so for example, we have a generation time distribution, so we think that most people, transmit on day 4 or day 7, and by day 10 hardly anybody's transmitting.
Mm-hmm.
And in some way your branch lengths sort of represent that, because it's a time until a transmission event.
Yeah, so
we're missing so much of the data.
Yeah, so what we've done is you can take if you're prepared to be deterministic about these things, you can write down a partial differential equation for this. And you essentially, there are probably better approaches, but the way that we've looked at it is by looking at an age of infection model. So with that, you can plug in all sorts of things in terms of the infection distribution, and then the coalescence ends up being a boundary condition. So when you're at the boundary of when the infection age is 0, then you have a choice of either coalescing or being an invisible transmission. So you can write down a PDE that describes it, and then you can play around with those generation times. It has the most pronounced effect, obviously, at the tips of the tree, especially if your sampling is biased towards a particular stage of infection, because you end up being very far away from the sort of stationary distribution, and that process gets more towards that as you go further down the tree. But it still does have an impact. So Trevor showed some simulations showing that differences in infectivity can generate differences in generation time. But if you have a fixed mean and then you change the variance, I don't think that too much it has too much of an effect. But the PDEs are a bit clumsy to deal with. So I've done simulations and shown that wrote down the models, showed that everything works in that context. So we can plug in non-exponential distributions, which we know are important for epidemic dynamics and so on.
So one thing we've got to do is add this stuff to our epidemic models. Going the other way, kind of how mathematically sophisticated is this phylogenetics, or is it just kind of the case there's loads of extra realism sat there and people can't include it in their inference process, or is it just that it's kind of not been a bit ignored by mathematicians, or what would you say that?
A bit ignored. So there's the simple idea of running things backwards in time. That was Eric's idea. He was a postdoc in UCSD, and I conjecture that pretty much any statistic that you can work out through either going from the tips of the tree down to the root, or vice versa, you can put into these models. So, for example, if you've got a source sink population, let's say you've got a reservoir, a wildlife reservoir, and you've got a human population, so you think the infection is going from one to the other, you can go back in the tree and you can count the number of times, just parsimoniously, you think it's jumped species. You can relate that to what the actual contact rate is in your model between those two species. And it's very simple bookkeeping. If you start to look at models where there's lots of structure and things can move around, then you have to sort of go backwards and forwards between forwards and reverse time. It's just a bit of bookkeeping. So mathematically, it's not particularly challenging. It's just a bit fiddly. So it's very straightforward, and there are sort of recipes. You know, you can. It doesn't require, you know, any fancy symbolic mathematics or anything. It's just a matrix equation that you can stick in. So if you are a compartmental epidemic modeler and you ignore networks, then you can do this stuff very easily.
Is anyone not ignoring Netflix?
Yeah, me too. Yes, it's awfully inconvenient. So, so you can probably get away with, you can do some approximations to do that, and they work okay. So, for example, you can take Eric's edge-based network models, and you can do the same thing. You can just plug in, what is the infection rate that comes out of a network model, then plug it into the same framework. It doesn't do too badly. It is a hack. It would be nice to know what the margin of error is there and what sort of implicit assumptions you're making when you're doing that. But does it work? Yeah, it works okay. So, just, you know, I can ignore networks, but I think it's still, if the data were there, it would be better. But it's just a matter of, it's another thing that you have to extrapolate from the samples of the population. And as Martina discussed, most of the network models have assumed that you have a sense of a network. Stuff on samples of networks is much, much harder. So you combine that with a sample of a phylogeny, or sample of a transmission tree, then you end up with something really horrific. Okay. Well, thank you everyone.